Hello, everyone. Welcome to today's broadcast, Efficiency for Validation, Continuous Change and Improvement. I'm Rita Peters, Editorial Director of Pharmaceutical Technology, and I'll be the moderator for this event. We're pleased to bring you this webcast presented by Pharmaceutical Technology and sponsored by Samsung Biologics. I'd like to share a statement from our sponsor. Samsung Biologics is a fully integrated CDMO offering state-of-the-art contract development, manufacturing, and laboratory testing services. With proven regulatory approvals, the largest capacity, and the fastest throughput, Samsung Biologics is an award-winning partner of choice and is uniquely able to support the development and manufacturing of biologics products at every stage of the process, while meeting the evolving needs of biopharmaceutical companies worldwide. For more information, visit samsungbiologics.com. I have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit your question by typing it in the Q&A box, which is located on the right-hand side of your screen. You can enlarge a slide window by clicking on the small square icon in the upper right-hand corner of the slide window, or you can hover your mouse over the lower right-hand corner and drag the window to the desired size. The slides will advance automatically during the event. If you have any technical problems viewing or hearing the presentation, click on the question mark Help widget in the dock at the bottom of your presentation window. Now I'd like to welcome today's speaker. Ray Cox is the Senior Director of Quality Engineering at Samsung Biologics. Thank you for joining us today, Ray. I'm going to turn the presentation over to you.
I'm going to talk today about the efficiency of validation and how we can do continuous change and improvements for the validation industry. Just to give a recap on validation, what is validation? So for those that are new to validation or have experience, it's documented evidence which provides a high degree of assurance that a process sequence consistently produces a product that meets specifications and quality attributes. So validation could be anything from the utilities, the facilities, the equipment, anything, the process, anything like that falls into validation. We want to make sure the system works properly and the process is reproducible. This is not a one-time event when we talk about validation. It's always continuous and continues on through the life cycle of the facilities and equipment. It's a regulatory requirement. Validation is important. Most regulatory agencies, when they do a pre-approval inspection, they come on board, they come onto a site, they look at have you done validation on your equipment, your processes. Without a successful validation, a company will not be allowed to produce pharmaceutical products, so it is part of the GMP guidelines. For the facility I'm currently working for, we follow the ASTM 2500 risk-based approach to validation. Lots of companies are moving in this direction. This is probably becoming the industry norm for validation. It follows a science and risk-based specification approach. And what we'll do is we'll start leveraging in the beginning for validation. We leverage across even starting at the factory acceptance level of equipment to the site acceptance all the way through the performance qualification. So different steps can fall at different places of what you're validating. So in the validation strategy world, every facility starts with the user requirements. And it says, I want a 15,000 liter bioreactor, and this is what I'm going to specify for the user requirements. And then we'll have the quality risk assessment, and we look at what the design risks are. Are we following everything we asked for in the user requirement? Then we have the further specification and design of the equipment, and then we get into the verification testing, which verification can be, it's almost the same as validation, and it's testing in combination. So it's, again, it's what I spoke on earlier. It's the factory acceptance test, the site acceptance test, commissioning, the installation operational qualification, the performance verification, and all these things in the past history, originally we would do each one of these individually. So sometimes for a lyophilizer, freeze dryer, we would do a full factory acceptance test, then we would bring a lyophilizer on site, install it, do a full site acceptance test, and then repeat all those same tests again as part of a installation operational qualification. And now, as we've gotten efficient in the recent years, we leverage some of those same testing. So if I was doing alarms and interlocks in the site acceptance test, there's no need to repeat those same alarms and interlocks again in the installation operational qualification. And once we get through the validation of these equipment, we have the acceptance and release testing, and which is just your basic summary reports where we're summing up all those validations, or we're doing summary reports for each one of those validation steps. But then we have an overall summary report that says, it has the trace matrix. Did I validate everything that was in my user requirements, and did I get what I asked for? Most people have seen the validation life cycle, so it goes through the same steps where you have the user requirements, the quality risk assessments, and then we go into all the validation where we have the performance verification testing, and then we get into the GMP operation. So once it's turned over to a facility, we're going into GMP operation. It falls under change control, and then the change control, if anybody changes anything, we're revalidating that equipment or doing it per addendums. And then we have ongoing validation, such for sterilization, where we do the SIP on a yearly basis, those kind of things. And then when you end your life cycle, you have retirement. So that's just a validation life cycle. So we have the continuous state of validation. You have change control. Once it's validated, it continues on, and you maintain it in a validated state. You have calibration, preventive maintenance as part of your ongoing maintenance of your systems to make sure your systems are staying in a state of control. We have your routine monitoring of facilities and utilities, so your environmental monitoring for your facilities. You have your monitoring of your water systems, your compressed gases. And then we have for time-based revalidation, which I hit on earlier, was you have your sterilization validation for an autoclave sterilizer you're doing on a yearly basis. For places where you claim sterility, you're doing on a yearly basis, such as depyrogenation tunnel, isolator, sterilization of a bioreactor, a fermenter, and doing those on a yearly basis with thermocouples and biological indicators. And then we have the periodic reviews, which are looking at the cumulative changes. So if there was looking at those on a routine basis, or is your facility or is your equipment still in a state of control? Has anybody made a lot of changes to it? Have there been a lot of deviations? Those kind of things where you're looking at for periodic review. So today, the real meat of this presentation, we want to talk about, we've always had the traditional validation approach, paper-based. And then, so each one of these previously, and it depends on where the state of a company is, some people may be still writing individual protocols and then routing them. We have a protocol for each piece of equipment. You take time to generate that protocol and route it for review and approval. You have the paper-based protocols that route in sequential order. They cause you to wait on the reviewer, and sometimes people have actually lost the document, or we don't know where it's at as it's routing around a large facility who has the protocol. And then we have simple protocols such as a controlled temperature unit, say a -70 freezer. Typical validation employee takes 4 to 8 hours to write a protocol just for a freezer, and then 2 to 4 hours for each one to review that protocol. And it depends on how many signatures you need review and approval of a -70 freezer. That could be 4 signatures times 2 hours, 8 hours. So what we want to talk about today is it's not a new concept, but master protocols and pre-approved forms. Different companies have used master protocols in the past and forms to do routine maintenance or validation. I hit on the -70 freezer. If you were buying 20 new -70 freezers, why do you want to generate a protocol for each one? And there's companies still doing this practice where each one, the validation technician is writing an individual protocol. They're putting in the serial number, the equipment number, and then routing that document separately. So what we want to talk about for, say, this -70 freezer, we can create a master protocol, which is pre-approved. It's been through all the signatures, signed off. What we want to do is take that master protocol and print it out and execute. And that same master protocol is used for all 20 of those -70 freezers. So you could validate all 20 at the same time without having to route a separate protocol for each one. So using a master protocol, they're consistent. There's no more arguing over wording and content. Past history, people have generated protocols, thought they had their template perfect, gained approval by all the signatures, and then the next protocol comes along and they see a typo or something they want to change. Even though it's the same protocol, they argue over minor details, route that, and change it again. So that's when you get to the master protocol level, you're not going back and forth with your quality assurance groups, your equipment owners, trying to get wording proper in your protocols. So for new facilities, currently working on a $2 billion facility, and we're going to use master protocols for everything. So we're actually going to use them for our factory acceptance testing. We're doing it for our site acceptance testing. We're doing it for our installation operational validation and our performance verification. So for a new bioreactor, if we're buying 10 bioreactors, we're doing one master protocol for a factory acceptance test. These 15,000-liter bioreactors, they all look the same. They're all built the same. They're all equivalent. That we just print out that same pre-approved factory acceptance test, go out and execute, and come back. And we're doing, so that gives you the idea of what master protocols are. It's just pre-approved protocols ready to execute. It cuts out that pre-approval step. For revalidation, some of the things we've done previously is you could do master protocols for that. So if I'm doing an autoclave on a yearly basis, we can generate a master protocol that says, okay, these are the steps I'm going to perform every year for my autoclave validation, where I'm doing the Bowie-Dick test, the leak test, I'm doing an empty chamber, I'm doing a minimum load, maximum loads, and they're already defined in that protocol, and I print that protocol out and go execute. The other thing is you could do it by forms. So each one of those steps in that, instead of having to generate a protocol, I can have pre-approved forms. So if I have a bioreactor and I know I'm going to install thermocouples and do a temperature mapping study with biological indicators for that bioreactor, I can have a pre-approved form that has all that information already there. And what I do is just print those forms out and go execute. Again, when you're doing revalidation, that's one of the things that is hard to get into the production schedules, and also to get time with the equipment. So you want to be able to just be able to print these and be ready. This one gives you an idea of a new facility. This is the $2 billion facility I'm currently working on. This gives you an idea of all the documents. And by the time we're done with this facility, it's going to be 8,216 documents just to get through the performance verification testing. And like I said, we're going to try to do all these with master protocols, trying to cut out any of that pre-approval, one approval and execute. So it goes into each one of these items of looking at the engineering, the equipment impact assessment, the URSs, all the way through the validation summary reports. So it depends on where you are in your validation life cycle. There are always ways to be efficiency in validation. The paper-based route, the review and approval in an electronic system. Most folks, depends on how large your facilities are or the capital investment you have. There's systems like Viva, those things that you can route your protocols through to be more efficient instead of having the paper-based. Again, the use of master protocols and pre-approved forms is another way just to be efficient. That way you're not routing multiple documents of the same type items. And then you have the paperless validation, which is the future. And a lot of large facilities are going in this direction. So once you have your templates defined and you know what you want your validation to look like, paperless validation is the way to go. It's all about the hardest part is getting that set up in the beginning with your templates. And then obviously using paperless validation is tablets on the floor, laptops on the floor. You're out there doing P&ID walkdowns. You're checking boxes on your protocols electronically. And you can take snapshots of pictures and upload them and do all that stuff to show you verification that it was performed. So how can validation be more efficient? Risk-based approaches. Switching gears, we want to look at anything where we're doing revalidation. And it depends on where you are, again, in your life cycle of your facilities. You could perform more of a risk-based approach. The things like the controlled temperature units. Some companies are executing a freezer on a yearly basis, doing revalidation. Some are doing 2 years, 3 years. Some are doing nothing at all. And it depends on what your risk you want to take. If you do this by do a risk assessment and say, okay, my -70 freezer, I have multiple building management sensors, thermocouples installed that are continuously monitoring my freezer for 24 hours a day, 7 days a week, alarmed and monitored, and my system is maintained by calibration and maintenance. Do I really need to revalidate a freezer on at all when I'm monitoring these systems? So it depends on what your risk is. It may be a risk to your product. So maybe if you're storing final product, maybe you do need to revalidate it more often. But maybe if it's something that's an intermediate storage, why revalidate? Same thing can go for cleaning validation. Some facilities are doing cleaning revalidation, cleaning monitoring programs. You could do it on a yearly basis. You could depends on if it's direct product, indirect product. Maybe you want to do indirect support systems such as buffer, media, those kind of things. on a 2-year basis. And then keep your critical cleaning revalidations on a yearly basis. And it's all based on risk. Do your FMEAs, doing any kind of risk assessment. So the other thing is risk-based. I hit on multiple large facilities where you have the same type of equipment. If you have 10, 15,000 liter bioreactors, matrixing those, why do SIP validations, sterilizing in place 3 times on each one, where I can do maybe 3 on one and 1 check on the other 9 vessels. So, and that can apply to equipment or SIP, can apply to cleaning, anything like that. Use and make sure you have equivalency documents written somewhere for these to say that, okay, all my 10, 15,000 liter bioreactors are the same. They've all been constructed the same. They're all the same size. They're all made of the same materials. And it's good to have those equivalency documents in place to support any time when you're matrixing. One of the other things we're looking at is dirty hold times. This is when you're doing cleaning validation, doing a dirty hold time for every production run 3 times for 72 hours, if that's your target for a dirty hold time. Doing that 3 times can be very time consuming and can be a bottleneck for your production. One of the things to do, again, is to do the risk-based, look at one dirty hold, holding at 72 hours for say, and it could be based on it's based on your knowledge of your products, your toxicity, those kind of things for cleaning validation. So it goes back to, again, why am I doing 3 dirty holds of these when I could be, if I'm following my risk-based approach, maybe I only do it 1 time. So in summary, change the mindset of the way you've always done it. Use pre-approved forms or master protocols for validation. So we want to be able to, for master protocols, we just want to be able to be ready to execute and consistent. So each one of these master protocols depends on how big your facility is. If you have tons of bioreactors, one master protocol covers all of them, and they're all consistent. So every protocol looks exactly the same. And then for your risk-based approaches, follow anything for your routine revalidation. It's based on your time intervals, take some risk. Look at your FMEAs, perform FMEAs, risk assessments, any way you want to do a risk assessment. Perform those and say, okay, I don't need to do this on an annual basis. I can do it on a 2-year, 3-year, or not at all, depending on what it's used for. So thank you. Hopefully this was informative. And again, thank you very much.
Unfortunately, we are out of time. I want to thank the audience for attending and for participating in this event. And I'd like to thank our sponsor, Samsung Biologics, for making today's webcast possible. Any questions asked during today's event will be answered via email. I'd like to invite everyone in the audience to participate in a brief survey, which you can see on the right side of your screen. Our audience members will receive an email alerting you when this webcast will be available for replay. I invite you to forward that announcement to your colleagues who may have missed today's event. Thank you again for joining us, and have a great day.